get into some good research institutes, some good research companies, like for example, like big innovator companies like Pfizer, GSK, Novartis, or even our good Indian companies like Dr. Reddy's and Ranbaxy. So if you really concentrate on research, and if you want to really get into research, study and do some thesis work during your graduation studies and master studies, and try to do, if possible, get into PhD programs in good schools, so that you'll have a better future towards academics also, as well as in the industries as well. And if you want to get into a pharmacist profession, better to do PharmD program. I think some of the colleges in India have started with PharmD program, which is essentially a 5 years course. So once you complete that, you will become eligible for working as a pharmacist as of now. Even rules may change, but there are some different opportunities nowadays. It's not like before, where pharmacy graduates used to paid less compared to software people and other people. So we always had that feeling inside. But now the things are different. We are like pharmacy professionals are highly paid, highly respected. So I think compared to even our old age, now you have better future and better prospects in pharmacy.
Tell me a few words about role of computers in pharmacy industry.
So now computation as such is called universal phenomena. We call it ubiquitous computers. That means computers everywhere. Whether it is pharma or drug manufacturing or hotel management or hospitality management or hospital management, computation and computers have a larger role to play. So when it comes to computational aspects, particularly in the pharma industry, gone the days of trial and error methods. Now the value of life has become very, very important. The value of life has gone much higher. No more we can experiment on the life. So we need to take the help of computation or computers when it comes to trial and error methods. We should say goodbye to trial and error methods and go for more precise and precision methods. No question of error. We want to avoid mistakes or errors, and that is possible with computation aspects.
Can you give some advices for the students, the present students?
Now, with bioinformatics, the word itself spells two different fields. One is biology, the other is informatics. We are trying to integrate two different fields and bring them together. That is sometimes called interdisciplinary or multidisciplinary approach. But we are looking much beyond those intra-disciplines. We do not want watertight compartments when it comes to the disciplinary approaches. We want to go for transdisciplinary approach. Transdisciplinary means across many disciplines at once and beyond single individual discipline. That's what we call it. So when it comes to healthcare and health informatics, or for that matter, disease dissection we call it, starting with diagnosis, through prognosis and cure. These three steps need to be understood very, very carefully. As I told you in my first question, we do not want to go for any error chance. We do not want to leave anything to chance. In fact, if you look at the present medical knowledge or discipline, we are leaving it to hope and chance. We do not want that to happen. We do not want to leave anything to hope and chance. We want to bring as much accuracy as possible. We want to make it as much measurable as possible. In fact, Socrates said, unless you measure, it cannot be called a science. So we want to get into the measurement aspects. That is what we call computation. So bioinformatics have a larger role to play in these aspects. That's why bioinformatics is very, very important.
I would like to really thank the organizers for putting this together. Praveen and rest of the organizers have really done a humongous job of trying to put together this nice conference. Very good to see you all. Thank you very much for inviting me. So this is the outline of my presentation. I may not go over all of them, but at least I will just have a slide or two of the bioavailability and bioequivalence, and then we'll talk a little bit about the modified release products and some of those challenges of modified release products in terms of bioavailability and bioequivalence. These are some evolving issues that I'll talk to you about. The locally acting products, I will say a few words about locally acting products, the highly variable products. I have seen Sam Hyder's presentation, and Sam is really a champion of this highly variable drug. He's done a lot of work, so I will defer this to him. I'll just have a slide or two, but for the rest of the presentation, please listen to Sam Hyder's presentation. He will have a very good explanation of the number of samples required and those kind of things for highly variable products. Now, I will talk about the dose dumping. These were some emerging issues. We still don't have some good solution for some of those problems. I will talk a little bit about dose dumping with alcohol. And finally, the target product profiles. I'll just briefly say what they are when I go to that slide, and how we can utilize some of those bioavailability studies to design a product to achieve the desired bioequivalence up front. Okay, so this will be the outline of my presentation. Now, the first couple of slides, please bear with me. I normally don't use very, the CFR related, very regulatory language, but to put things into perspective, I have pulled a slide where the definition for bioequivalence is there. So at this time, let's say it's a bioavailability and bioequivalence. What's the big difference? Really, bioavailability is like a benchmarking effort, and bioequivalence really is some comparative test. You have a product A versus product B, you're comparing it, or brand versus generic, you're comparing it. But in the case of bioavailability, you're trying to get some benchmarking of whether the drug really gets absorbed, whether you really have some kind of some rate and extent of drug absorption there, or how does it compare with your IV, what fraction of the drug get absorbed, or where does it get absorbed from, for example, your GI tract, stomach, duodenum, jejunum, if you're trying to get an estimate of that. So you're trying to get some ideas, or you're trying to have a benchmarking effort for bioavailability, and bioequivalence is an issue where you are really comparing your product to an RLD and trying to see whether your product is equal to them, right? So now in bioequivalence, this is a CFR definition. It's like a language, regulatory language that we have to see. So if you look at the CFR, Code of Federal Registry, the absence of a significant difference in the rate and extent to which the active ingredient or active moiety, there's a slight difference, active ingredient and active moiety. So you have the study, I briefly mentioned about the plasma concentration versus time. So you have the data, and then you analyze the data, right? So the data analysis, I think this morning somebody highlighted the importance of statistics there. And so you analyze the data. So in this case, you're saying that significant difference is 20%. So what you're doing is you're doing two one-sided t-tests essentially. So one of them, test T is not significantly less than the reference product. So in that case, the 90% confidence interval on test over reference is greater than 80%, right? Or sometimes, then you also do the 90% confidence interval on test over reference is less than 125%. So essentially, it is between 80 and 125% with a 90% confidence. So this has worked well for a long period of time, still works well. But sometimes, so when this study is done, the outcome of this would be, so this is what you'd get. So you did a study, right? So where does your product fall? It falls in one of these regions, right? So you might say, well, if this is the product, say this is between 80 and 125% of test over reference, so now you have demonstrated bioequivalence, be happy that it's done. Your product is, if it is pharmaceutically